I'm going to discuss about the regulation and targeting of androgen receptor nuclear localization in prostate cancer. And I would love to get your questions, suggestions after my talk. This is my disclosure. And you all know that the anti-radiation therapy is the standard treatment for men with metastatic prostate cancer. However, they all, almost all of them relapse. And the second generation AR targeting agents such as abiraterone and enzalutamide used to treat these patients. And unfortunately, patients will relapse again. The mechanism for the resistance of AR targeting agents are complex, but major mechanism is the deactivation of the androgen receptor. According to literature, that about 70 to 80% of the patients that becomes resistant to these agents are still expressing TSA, indicating that the deactivated AR is still the driver of these resistance tumors. And targeting these deactivated AR remains to be clinically very important. Many years ago, Dr. Gregory and his colleagues published into Wang a very important finding that I read this paper. And what they showed is that the androgen receptor in castration-resistant C4-2 cells is predominantly localized to the nucleus in the absence of androgen. Whereas the parental androgen-sensitive LNCAP cells, AR is predominantly in the cytoplasm. And why this is important? Because AR is a transcription factor. Its activity requires this nuclear localization. So presumably, that androgen-independent nuclear localization of AR in CRPC is a necessary step leading to androgen-independent activation in CRPC. And my lab had generated the C4-2, the GFP tagged AR and transfected into TC3 link cap and the C4-2 cells, and confirmed that the GFP tagged AR is predominantly localized to the nucleus of C4-2, but in the cytoplasm of AR-negative TC3 and androgen-sensitive LNCAP cells. And this is consistent with publication of others. And of course, Barbara synthetic antigen can drive antigen into the AR into the nuclear receptor. So this raises the question, so what is the mechanism leading to this androgen-independent nuclear localization? And also, can we have some ways to prevent this androgen-independent nuclear localization? Because we think that if we can prevent AR goes to the nucleus, we might be able to inhibit the CRPC growth. So this is like the very straightforward thinking. So in order to understand the androgen-independent nuclear localization in CRPC, or the dysregulation of nucleus cytoplasmic trafficking of androgen receptor, that's what we were thinking at that time. It would be important to understand how AR is imported as well as exported in the nucleus. This is a classic model at that time, and it's being sort of being used in the field for several decades. So AR is made in the cytoplasm and the complex with chaperone is heat shock protein. And then upon DHT binding, the heat shock protein dissociates and ligand the AR goes to the nucleus to transactivate targeted genes such as GSK. Upon androgen deprivation, DHT can dissociate from AR, and unliganded AR was thought to be exported to the cytoplasm. And So this was the model, and the nuclear impulse signals was reported, documented in the literature. There are two of them. Nuclear impulse signal 1 and L1 is located in the DNA binding hinge region. It's well described. Another impulse signal L2 is in the ligand binding domain, but it's only present in the ligand LPD, so the precise location of L2 remains unclear. However, the nuclear export signals was not clear. There was some people claim they identified the nuclear export signal, but my lab was not able to confirm that. And so we want to search for the what the nuclear export signal was is located. So we all know that intracellular localization of AI is regulated by androgens. So we hypothesized that the nuclear export signal may exist in the binding domain of AR. So we made as a preliminary pilot study, we made the GFP tag NAR. It's a deletion construct lacks LBD, and then the GFP tag LBD. As you can see, NAR localized to the nucleus, whereas LBD is predominantly in the cytoplasm. So this suggests that NAS should be present in the LBD. And as expected, androgen, synthetic androgen boron can drive the LBD to the nucleus because of NL2 is present in the LBD. And once the LBD is in the nucleus, in the presence of boron, we can withdraw LBD by culturing the chocolate through the medium. And the cells, in the absence of androgens, the GFP tag AR becomes cytoplasmic localized. And even in the presence of protein synthesis inhibitor, they also becomes cytoplasmic and localized. So this data was reproduced just by my lab, but any other lab in Pittsburgh as the nuclear export signal drives that. That was actually later on we figure out it's very naive thinking. There are alternative interpretations for this observation. But regardless, with this interpretation, we decided to identify the localization of nuclear export signal is using N-terminal deletion mutagenesis. We defined the N-terminal boundary of NES to the N3 position, and the C-terminal deletion mutagenesis analysis located the C-terminal boundary of NES to the C6 position. And this gives the NES the position of 743 to 817 of AR. So this is a rather large nuclear export signal. Because classic traditional nuclear export signal is less than 10 amino acids. That's also leucine, which are conserved amino acids. And that's not present in this NES. So this suggests that the mechanism of action is very different. And as expected, the NES, also AR, also like the binding domain, its cytoplasmic localization is resistant to leptomycin B, which is an inhibitor of exporting 1, also called CRIM1. And that leptomycin B can inhibit the classical NES from protein kinase inhibitor, as you can see that. So this suggests that the mechanism further indicate that the mechanism of export by this putative NES is very different. And in fact, my lab have tried for a long time to identify the factors that support mediating the nuclear export of AR and failed. And there are people published some papers that they identified the exporting factors going up, but now we are going to confirm. We really spent like about 10 years to search for that and couldn't find it. While we were searching for those exporting factors, we also noticed that very important phenomena, that is that any constructs that are tagged with AS, AR from AR, the expression level is so much lower. And this suggests that this might be a degradation signal. So we measured the half-life of AS containing fusion constructs. As you can see, the half-life of GFP tagged AS is less than 6 hours in the presence of protein synthesis inhibitor cyclohexamide. And similarly, the GFP NAR-HS also is very much unstable, whereas GFP and GFP-NAR is very stable in 24 hours. It's not it's maintained a pretty good level in the presence of protein synthesis inhibitor. And another method is that we use is S35 methionine pulsing in the panel C, and you can see that the half-life of GFP and S is less than 3 hours, whereas the GFP is very stable, even after 32 hours. So a lot of proteins left. So this suggests that this NASAR is a very potent, robust protein degrader signal. And one of the major mechanisms causing protein degradation in cells is the ubiquitin proteasome system. So we explored if the NASB protein degradation is also mediated through this system, the UPS system. And using MGM32, which blocks the proteasome activity, we showed that the degradation of NAS tagged fusion protein was significantly inhibited, as you can see in both the panel A and panel B, in the absence of presence of inhibitor 32 at different doses or different time. And as expected, the NASAR can promote polyurination of the tagged protein, as you can see here. So this suggests that the NAS actually promotes protein degradation through enhanced urination. And so this raises the possibility that we were thinking that maybe NAS is not an NAS at all, rather it's a degradation signal. So maybe it's causing very robust, very rapid degradation of the protein in the nucleus. So one trainee, Dr. Zhou, he used the various constructs and showed that for the constructs containing NAS, when you treat cells with MGM32, and all these experiments are in the presence of cyclohexamide, block the new protein synthesis, you can see a shift of the signal from the cytoplasm to the nucleus, whereas without NAS, you don't see that. So this suggests that the NAS can enhance AR nuclear degradation. So maybe NAS is a nuclear degradation signal. And because I don't have a pointer, so I cannot really point at since the location screen excuse me for that. So this raises the possibility that the important AR in the nucleus is not going out. Instead, it's rapidly degraded. And this actually is shown here in this experiment. These are the experiments in the presence of new protein synthesis inhibition, inhibit CHX. And when we withdraw the androgen after the GFP was driven into the nucleus by DHT, you can see the signal in the nucleus significantly reduced in the COS7 and the CT cells, but not in C4-2 cells. The C4-2 cells is a CRTC suggests that the proteins in the nucleus is more stable in the C4-2 cells. And when we treat cells with Androgen-32, the nuclear degradation was blocked, but you don't see the nuclear signal goes out of the nucleus. So this suggests that the nuclear, important nuclear AR is degraded, not exported following DHT withdrawal. And we also cross-labeled the AR using Viash. That's a small, it's a compound that labels tetracycline tagged GFT-AR. So the red AR is cross-labeled, the green AR is the total AR, and it's both mucin size as well as cross-labeled. And in the absence of Androgen-32, you can see the cytoplasmic localization of GFT tagged AR in COS7 cells. And but the cross-labeled AR, once in the nucleus, it doesn't come out. It remains in the nucleus or degraded. And in the presence of Androgen-32, it blocks the degradation of cross-labeled AR. And a similar situation observation was found for RNCAP cells. For C4-2 cells, again, the cross-labeled AR is more stable. But regardless, we don't see the red AR goes out of the nucleus, remains in the cytoplasm. And another method that shows the same thing. We cross-label the AR, endogenous AR, with HPG, which is synthetic, an analog of methionine that can label the endogenous, cross-labeled endogenous AR. So the bottom line that the cross-labeled AR in the cytoplasm does not increase following the androgen withdrawal. Its level only decreases. So there is no any evidence for the endogenous AR to be exported. The AR level, the cross-labeled, does not increase of the click-labeled AR. It's a little complex. Without the point, it's hard for me to point out which you have to trust me or read the paper for details. Then another thing that from this observation that we believe that AR can be imported into the nucleus because in C4-2 cells we know that in the absence of androgen, AR still goes to the nucleus. So that suggests that androgen AR can undergo an independent nuclear import. To prove that, we generated a GFT tag mutant AR L859F. It's a non-androgen insensitive AR mutant that is not able to bind to GFT. So as you can see, in androgen-sensitive cells, DHT does not drive in the nuclear localization of this mutant AR. However, if we block the protein degradation, there is a shift of the cytoplasmic signal towards the nucleus to suggest that the AR can indeed undergo nuclear import in the absence of androgens. And of course, C4-2 cells, in the C4-2 cells, we see the same situations, observation on different culture conditions because nuclear AR degradation is compromised in C4-2 cells. So this provides a very strong evidence for androgen-independent nuclear import of AR. And because AR undergoes such a robust degradation in the nucleus, we tested if the AR undergoes ubiquitination more predominantly in the nucleus. And actually, it was very surprising to us that in both RNCAP and C4-2 cells, AR ubiquitination was pretty much only detected in the nucleus. AR ubiquitination and degradation has been studied extensively, but it was not known that it only undergoes ubiquitination in the nucleus. So one of the important questions my lab wants to understand is why this happens, how this happens. And also suggest that the nuclear AR ubiquitination is more profound in NCAP than C4-2. That provides an explanation why C4-2 cells AR is more stable. And one important AR-E3 ligase that puts ubiquitin on the AR is MDM2. There are 7 or 8 AR-E3 ligases, and because of limited manpower, we picked this one because according to literature, this appears to be a major one. Since you knock it out or knock it down, AR becomes more stable. So it has to play an important role, a major role in AR stability. And what we show here is that this co-IP experiment suggests that MDM2 only associated with AR in the nucleus in both LNCT and C4-2 cells. This explains, in part at least, how AR is specifically ubiquitinated in the nucleus. Then the next question is why it only associates with AR in the nucleus. So that's a question that we are addressing at this point. And then, as expected, knockdown of MDM2 reduced the nuclear AR ubiquitination, and also knockdown of MDM2 enhanced the nuclear AR level. This is all expected results. And we all know that androgen can enhance the AR stability. And this suggests that one of the mechanisms that androgen binding to AR can repress nuclear AR ubiquitination as a mechanism. So based on our study, we propose an updated model for AR intracellular trafficking, which is really a one-way trip. So AR not only can undergo androgen-induced import, but it can also undergo androgen-independent nuclear import. And also importantly, that imported AR does not undergo nuclear export. That means we do not need to search for exporting factors for AR. And also, we show that NES is a signal for AR ubiquitination and degradation in the nucleus. So we think it's more appropriate to call NES as a nuclear degradation signal, as NDS. And also, we have some preliminary data which needed to be confirmed further with different models that AR nuclear localization in CRPC cells is likely due to the reduced AR ubiquitination and the degradation in CRPC. And these are all testable hypotheses. So while my lab was studying these basic mechanisms regulating AR nuclear localization, we were also trying to explore the possibility that blocking nuclear AR localization, because we believe that if we have small molecules that can block AR nuclear localization, that could lead to potential treatment for CRPC. And I was very lucky, had a very strong collaborators, Dr. Zhou Nelson. Many of you probably know him. He's a urologist scientist and with extensive experiences in clinical trials in prostate cancer. And also Dr. Peter Whiff, which is really essential. He's a very outstanding medicinal chemist. And we first collaborated with Dr. Paul Johnson, who is an expert in high throughput screening. We developed a ARGMC type fluorescence microscopy assay. It's a high throughput, high content assay, and screened over 200,000 compounds. And the cherry picked 2,500 candidates, and then over 300 was confirmed to be active. And out of that, 120 had IC 50%, 10 micromolar. And then we sit down together with structural biologists and medicinal chemists and triage the majority of this compound. And eventually selected about 23 of them for further testing to confirm this activity. And this algorithm that we use to for the high throughput screening. So the nucleus of the prostate cancer cells defined by staining, and then the computer will draw, the software will draw a yellow circle mask for the nuclear AR level and a red green mask for the cytoplasmic level. And this is actually one example of how the software did for the localization of AR. And out of those 23 compounds we tested, 3 of them appear to be very interesting because they 5 and 9 cause cytoplasmic localization, and compound 10 significantly reduce the AR level in the nucleus. And also, these 3 compounds does not cause cytotoxicity of cells. Cells are okay. We know blocking AR shouldn't kill cells. Any compound that kills cells will eliminate them. So these 3 compounds were further tested. And interestingly, the 2 compounds, 5 and 9, they are pretty much the same, very closely related. So we presume the mechanism of their action should be the same. And we confirmed again that it can cause cytoplasmic localization and was able to inhibit AR target gene TSA. And importantly, one of the compounds we in the animal model of relapsed LINCAP tumor, it was able to significantly inhibit tumor growth. And this compound, CGI, was also causing cytoplasmic localization of AR in C4-2 xenograft tumors. And of course, it inhibited the Ki-67 index in this tumor model. Since we know that CGI can cause cytoplasmic localization, and so we tried to see what's the mechanism. It turns out that it was able to enhance the nuclear degradation. As you can see, the pulse labeled red AR was significantly reduced following the treatment with CGI. In both LNCT and C4-2 cells, and this induced the nuclear AR degradation by CGI can be blocked by Androgen 32. So further, we tested that the CGI actually can enhance the nuclear AR ubiquitination in C4-2 cells, and we're very lucky that showed that CGI can enhance the association of AR with MDM2 in the nucleus. So presumably, this is the mechanism, at least in part, the mechanism that causes the efficient AR nuclear degradation in C4-2 cells by CGI. And when we knock down MDM2, it partially inhibits the CGI-induced nuclear degradation, suggesting that there might be other mechanisms that are induced by CGI to cause nuclear degradation. And interestingly, we showed that CGI can also block AR nuclear input. And in this SCOT7-cell model, you can see that control without any antigen is in charge the medium. In the presence of Androgen 32, the AR can have a shift from the cytoplasmic to the nucleus. And of course, in all these experiments, they were treated with cyclophosphamide to block the nuclear synthesis. And so this provides additional evidence for the androgen-independent AR nuclear localization. And the CGI was able to block it. And we tested the enzalutamide or MDB-3100 because there was a paper saying that MDB can block the nuclear localization trend. But in this experiment, under these conditions, we see actually the MDB promotes the nuclear localization. So it's one possibility maybe MDD can promote the AI degradation in the nucleus. Because a lot of the previous experiments did not include enzyme to block nuclear protein degradation. So further studies show that CTPI can compete DHT binding, so suggests that it is a capacitive inhibitor. And using purified AR, we showed that CTPI can bind to AR directly using the surface plasmalosis STR assay. So suggesting that this is a direct inhibitor, direct binder. And use computer modeling, we found the two potential binding sites of CTTI in LBD1. The higher affinity binding site is at the position where DHT binds. So that can explain why CTTI can competitively inhibit DHT. And we're collaborating with structural biologists to define how CTTI binds to what kind of conformation. So based on our finding, we think that the CTTI can inhibit AI activity via a number of different mechanisms. One is that it can block, unlike the AI nuclear input. And the two is that it can compete with DHT for binding and block its nuclear input, and also can enhance the recruitment of MDM2 to promote nuclear AR utilization. Although this is a very interesting small molecule for our research, and also potentially can be developed for future therapeutic usages. But our medical science medicinal chemists think that this compound 10, is more drug-like based on its structure. And at the 2 micromolar, this Western blot, you can see it can inhibit the TSA level without significantly inhibit the AR level. So suggesting that it can inhibit the AR functions at a lower concentration, but at a higher concentration, 10 or 15 micromolar, it was able to significantly reduce the AR protein level. So it can act through different mechanisms to inhibit the AR. And this TSA-driven luciferase assay, we used it to show that compound 10 can inhibit the AR function. And also the endogenous TSA was inhibited by this compound at 1 or 5 micromolar, pretty low dose. And compound 10 in this slide shows that it was able to inhibit proliferation of AR positive but not AR negative prostate cancer cells. In fact, we tested additional cell lines, and this conclusion is still true. And one thing I'd like to point out is that the 25E1 that express AR7 and is resistant to glutamide can be inhibited by this compound. And this shows the control experiment that the MDD does not inhibit the 25E1. And the compound 10 was able to inhibit 25E1 xenograft tumor growth. So that suggests that it could has a potential for further development. And it prolonged the survival of mice bearing 25E1 tumors. And in contrast, this compound did not inhibit the PC3 tumor. And our medicinal chemist initially made 67 compounds by altering the structure of 4 different zones of this primary molecule. And one of the compound, number 43, was able to be conjugated to agarose Ds. And we tested that after adding the linker to this molecule, it was able to retain the activity of this small molecule. And this compound 10 analog 403 conjugated beads was able to pull down GFP tagged AR, but not GFP tagged RB1. And the pull down by 403 can be competed by compound 10, so suggesting that there is a direct binding of androgen receptor with compound 10. And the endogenous AR was also put down by 403 and can be competed by compound 10. And more importantly, purified AR can also be put down by 403, but not by the control. And one of the analog compound 403 has a two racemic form, and this mixture of molecules was able to inhibit the PSA luciferase in the luciferase assay in the presence of androgen in C4-2 cells. It's slightly less effective than MDD 3100, but it's comparable. And also, this compound inhibited the transcription activity of AR deletion constructs, the NAR we call. This AR lacks the LBD. So this suggests that this small molecule can target the N-terminal domain of the androgen receptor, and it can be used to inhibit the ARV7, AR variants expressing prostate cancer cells. And we also did the RNA-seq analysis. All the compounds were able to significantly, to pretty much block the AR transcriptome induced by RAKD1. And a minus form of the JJ450 can behave pretty much like enzalutamide in terms of inhibiting the androgen response genes. They are almost like over 99% overlap with the enzalutamide. So suggesting that this compound can be quite specific to AR. And Dr. Pascal showed that with this compound, either administered through oral gavage or intraperitoneal injection, was able to inhibit the relapsed LNCT xenograft tumors, suggesting that this compound can be orally bioavailable. So it's a good sign for further development. And also another analog which we use is JJ74138 at the 10 milligram per kilogram IP injection can inhibit the growth of turnitin RB1 tumors. And also we used the SPR, surface plasmalosis, showed that this analog can interact with AR directly. Furthermore, in the tritium-labeled DHT binding assay, this in the L95 cells, this compound did not inhibit, compete with AR. And there might be a little bit of competition at a very, very high dose, but at the dose that we used, it's not. And also, recently we showed that this compound was able to specifically or preferentially reduce the AR level in the nucleus of C4-2 cells. So we did additional experiments show that it can block AR recruitment to androgen responsive elements, which I'm not showing here because of the time limit. But in summary, we think that this compound and its analog is a very interesting small molecule for further development, and it can directly target AR as well as AR variants that are lacking LBD. And it seems to be very specific because it inhibits the AR positive tumor cells, but not the AR negative cells. Every tumor cell line we test AR positive cells was inhibited. And the toxicity seems to be very low because we can give I think the highest dose we give was 350 milligrams per kilogram in animals and did not detect active toxicity, acute toxicity. But we cannot deliver any further because the vehicle will kill mice. So we don't know what's the maximum tolerated dose yet for this compound. And also, I didn't show here, we had some unpublished data suggest that this compound can synergize with enzalutamide, which is actually expected because they're targeting different regions of AR. For the future studies, we wanted to continue our exploration to define the mechanisms and also factors responsible for nuclear-specific AR urination and how these nuclear-specific urinations dysregulate the CRPC, and if we can block that dysregulation. And also, very importantly, that we wanted to develop more PK pharmacokinetically suitable, more stable, and higher bioavailability with higher bioavailability compound to inhibit the enzalutamide resistant CRPC clinical trials. And this will be a major challenge. And also, we want to explore the potential synergies with enzalutamide with this kind of compound. And also, we're trying to collaborate with structural biologists to determine the mechanism of the action of these novel AI agonists. We don't know where they bind, how they bind, what kind of structural alterations triggered by these antagonists. So we have a lot of things to do in the future, in the near future. And I'm very grateful for the people that have worked in my lab, and they made a significant contributions that we can see. And also my collaborators, as well as the funding from UPMC. And with that, I'm very happy to answer any of your questions. Thank you.
That was a wonderful talk. Thank you so much. Very exciting, to see sort of take the concept into drug discovery and development. I had two questions. One about the role of exogenous testosterone and testosterone in general in this. Have you thought or looked at the role of testosterone sort of modifying or modulating AR expression and the AR pathway?
You mean the exogenous testosterone, not DHT? Not DHT.
And the other question was DHT.
Yeah.
There is sort of this interest in bipolar androgen.
Yeah. Actually, it's another area I have worked at for a while on the T versus DHT. It's a fascinating thing, but it wasn't my focus. Wasn't focused on the testosterone regulation, exogenous testosterone regulation of androgen receptor. Because it's known actually published by quite a few different labs. Androgens can enhance the stability of androgen receptors, so at protein level, it can increase the protein level. But if it can suppress mRNA expression, there is a feedback, right? There is a mechanism for that. At least in some cell line models, that's what I know. Regarding the bipolar androgen regulation, that actually is a very exciting thing. And when I was at Northwestern, one of the things we discovered very interestingly was testosterone is more potent than DHT to induce some of the androgen tumor suppressor genes in the regressed castrate prostate. but not in the full-grown prostate. And this was very reproducible. Actually, it was discovered by Sam Davis. He was a PhD student, and showed that I was so surprised because that's totally unexpected. We still don't know why, but I asked him to repeat the experiment whether you shut up the samples, but that's no. And the bipolar androgen ablation it is a little different from that because we all know in most of the prostate cancer cells, androgen effect on cell growth is 5 phases, like LINCAP, LNCAP cells at the 0.1 or 1 nanomolar level is stimulus growth. At the very high dose, 10 of the ball is suppressive growth. And in the C4-2 cells, maybe the biphasic growth shifted towards to even lower level of androgens. So in the CRPC, so they will be more sensitive to the tumor cell growth, the high dose of testosterone, because the sensitivity to the growth optimal, those shifted to the lowest side. So that probably is what happens. And those concepts was promoted a long time ago. I don't know Dr. Kim might know. You know Dr. Sendell. He has actually promoted this for ages. A lot of people didn't believe that there. Because no one wants to do clinical trial because the patients, they said it's not going to happen because it's not happening. You can't get prostate cancer patients with high dose of testosterone. And not until Dr. Stanford Edwards, very great, very thoughtful, obviously, backed by studies to do those experiments. So I think that's another story that tells you that we need to be open-minded with androgen signaling. It's very complicated in different systems. It can behave differently.
And the other question was DHT. I mean, so it seems DHT is fundamental to the mechanisms that you showed. But why is it, do you think, clinically we don't see much effect with DHT suppression? 5-alpha reductase inhibitors really seem to have limited clinical value.
Okay. The reason we use DHT was because it's much easier to get that. If you want to get testosterone, a lot of times you have to go through drug control. And I don't think it matters. We can, in a lot of times, we can use either DHT or for a lot of studies. Because DHT is a sort of a weak form in most cases, although they have differential function from DHT, but they can do a lot of overlapping function with DHT. So you block DHT because it is still can drive androgen receptor. Most of the signals so that's why I probably don't see much effect.
Thank you.
Any other questions? I have a couple of questions, Joe. That's fascinating. So really intrigued about the lack of nuclear degradation in CRPC. So what do you speculate as a mechanism as to why that degradation arm is deregulated or dysregulated in CRPC? What do you have found so far?
Can you say it again?
So like in C4-2s, right? That in the nuclear degradation, because they become stable in the nucleus.
Mm-hmm.
So you speculate that the degradation in the nucleus does not occur or is dysregulated in castrate-resistant prostate cancer. So what have you found so far? What do you speculate as a mechanism? Is that MDM2 or what's going on there? What have you found so far?
In short answer, that I wish I know. I don't know in detail. There is some preliminary data suggest that at least one of the players is MDM2, but obviously it's very complicated. We haven't had a chance to check for the other important E3 ligands. And based on the several cell lines we have, it seems that the MDM2 block in those more aggressive, so like 29B1 and 95, the level is very low as compared to the lead cap.
In the nucleus, or is it your overall?
We check the overall,
Overall.
but in the nucleus, it also should be lower because in the just between C4H2 and RCP, it appears that's the case. And these assays has to be done in the presence of MDM2 because MDM2 can undergo auto duplication and rapidly degrade. You don't see them if you don't have the level is very difficult to measure. And if you check for MRE level, it's not going to tell you the whole story. So, but we plan to look at the MDMT and other PC ligands at different levels, and preferably with clinical specimens show that their level is decreased, which one is decreased. And I think it's going to be very complicated.
So in general, do all or most of E3 ligases have the nuclear import signal, or how do they get in there?
I haven't studied this question, but I believe that a lot of proteins can undergo nuclear import. There are a number of mechanisms. One is that if the size of a protein is smaller than 60 kilodalton, it can go through the diffusion mechanism because the nuclear pore size is big. Unless it's like a protein like AR, it cannot diffuse. It has to be energy driven with the impulse signal. And then you have impulse signal and another mechanism, a piggyback mechanism. If it binds to another protein that it can be imported, then they can be imported along with the other protein. So it's it can be challenging.
So the second question, so the compounds, JJ compound that you have, do you have any experimental data now? You've shown, I think, nice effects about how they overlap in terms of functions, in terms of inhibition with enzalutamide or MP3100. Any evidence right now, cellular evidence, that it actually is active in enzalutamide resistant cells?
Yeah, it does. Like in L95 cells and 25B1, it's active. They were active in cells.
Then one last question. The nuclear import of the ARP7, does JJ compound prevent that also?
We did not study the import of ARP7. It's actually another fascinating story that the current literature it's needed through some DNA binding of the AR, because ARP7 doesn't have a mutation. Mutation splice there, it's all the nuclear import signal one, so they don't really have an active nuclear import signal. So it's fascinating. We did not study that. We did not test the block the nuclear import, but it seems that it can induce nuclear ARP7 activation, although not as effective as full length AR. And importantly, that this JJ compound, it doesn't have to induce the activation in order to inhibit the activity, because all you need to find, even without the activation, still inhibits the activity.
All right, thank you.